All right, here we are for another AP biology video. The last video was about cell communication. We talked about the basic idea of cell communication, why it would happen, a lot of different variations on how it would happen. But one thing that was in the video was that all the variations lead you back to this video called reception, transduction, and response. In this video, we want to know more about all these different steps. So let's get right into it. We'll start, of course, with reception, the first step in the communication pathway. Now, here are the basics. Like I said before, we always have a signal molecule. It's got to be a physical object that then connects to another physical object, right? The signal molecule has a name called a ligand. You'll definitely see that word all over the place. Ligand is a generic term for any molecule, usually a smaller molecule, that binds specifically to another molecule. This is kind of like the idea of a substrate that binds to an enzyme. It's very, very similar, except that the proteins that we're talking about aren't technically enzymes in that sense. And so in this case, we end up calling it a ligand rather than a substrate. Now, the receptors can be membrane bound or intracellular, but we'll get to that in a second. First, let's talk about ligands, about signal molecules. Of course, they all have a very specific shape. Those shapes have to fit into the protein receptor, like a lock on the key. Again, very similar to an enzyme situation. The idea here, though, is that all the ligands have different specific shapes, right? Epinephrine has to be a different shape than, say, growth hormone, or else every time you wanted to release epinephrine, you would make your cells grow instead of releasing glycogen or whatever it is, right? Like there's every different signal needs a different shape. And so there's lots of different shaped signal molecules out there. There's also lots of different receptor proteins. Now, not all cells have the same receptor protein though, right? How is it that epinephrine can travel through your entire body, but only affect certain parts of your body? Well, of course, the muscle cells that need to receive the epinephrine so that they will turn the glycogen into glucose, they have the right receptor for epinephrine. But other cells, say like your pancreas, which doesn't have a whole lot to do during your fight with the mountain lion, it has different receptors because it's waiting for a different signal, right? It's not waiting for a fight or flight signal. It's waiting for something else that has, honestly, about to do with your digestion. And so when the epinephrine hits those receptor proteins, they just bounce off. And so those cells don't respond to epinephrine, but the cells that should respond to epinephrine do. Of course, this is all determined by the DNA of the cells, right? Only certain genes are active, are being expressed in different cell types. So the tissues, like muscle tissue, is going to have certain receptors, and then eye tissues will have different receptors, or pancreas tissues, or whatever it is, right? So it's all about the combinations, which is an idea that we've talked about before. Now, more about signal molecules. There are two types of signal molecules. They are shown on the screen here. Epinephrine is an example of one type. Testosterone is an example of another type. Can you tell by looking at these molecules what the division is? How do you group these two different molecules? What's different about epinephrine than testosterone? And the thing that's different about epinephrine than testosterone is that epinephrine has multiple OH groups and a nitrogen group, and it only has one ring. Testosterone has two O's, but it has a lot of rings, right? And so this one is tricky. You have to remember way back to the beginning of the first semester that having a lot of OHs is going to make you a polar molecule. Now, testosterone you might have thought was polar also because it has the O and the OH. However, that big section in the center with all the rings, and you notice those rings don't have any O's or N's, which means these rings are completely made up of carbon and hydrogen. This is a very big non-polar area. Way too big to be considered polar, considering you only have these two tiny spots of polarity right there, right? So even though this one, testosterone, has oxygens, it's only a couple, and it's largely non-polar. Now, why do we care about that? Polar molecules can't go across the membrane, whereas the non-polar molecules can. This is going to mean that we need different receptor proteins, right? If you have a polar ligand, you must have a membrane-bound receptor with the active site, if you will, on the outside of the cell, because the signal molecule will never actually get inside the cell, right? It can't pass through the membrane. Meanwhile, for For testosterone, it is nonpolar. It can just go straight through the membrane. Having a protein receptor on the outside would be a bad idea because it would just get bypassed. The hormone would just go through the membrane and miss the receptor altogether. And so you have an intracellular. Remember, intra means inside or within, not inter, which means across or without, right? So there are two different types of protein receptors. They all fit into, those are the two categories that many examples will fall into. Now, the membrane receptors get a little complicated. I'm going to go through some of them right now. There are three main types. You aren't responsible for memorizing those types, but you will come across them in the examples. And there is one common theme, a pattern that you do need to know. And that pattern is that when the ligand binds, we will get a conformational change. This is a pattern that we've seen many times before, actually, right? When a small molecule binds to a bigger protein, it makes the protein change its shape, right? And so shape is the way that biological molecules carry information. So the shape of the protein, in one sense, is going to be inactive. When it changes its shape, it becomes active, and vice versa. So the first one to show you is this one called the G protein linked receptor. In my experience, this is the most common one that I've come across. I don't know if it's the most common one used in say, human cells. But when I've been doing my research on various pathways, a lot of them start with a good old G protein linked receptor. Now, again, there is a pattern here. Step one, we have our, oh, awesome. Our system is ready to go. It's just waiting for a signal. Step two, the signal binds, which activates the receptor, reception. The reception then causes the G protein that was here to take in a little bit of energy to give GTP, to give some energy to this particular molecule. That makes it change its shape so that it can fly off and activate this enzyme. Eventually, the GTP gets used, the energy gets used, and everything goes back to where it was in step one. Now, again, you don't have to memorize this, but when you are looking at a diagram like this, you need to be able to follow the steps. Step one triggers step two, which triggers step three, right? I know it's 1, 2, 3 like that, 1, 2, 3 like this, but you can see it just in this first picture. Step one, step two, step three, right? It's all just leading to the next molecule. So that's one of them. Another one's called the receptor tyrosine kinase. You'll notice that this one looks definitely different. And this one's especially different because it needs two signal molecules at the same time, right? This would be a way for a cell to kind of guard against accidentally triggering the response. If you had one signal molecule that randomly just happened to find the receptor, it would trigger a signal. But this time, you need two signal molecules, and the chances that you have two signal molecules randomly floating when they're not supposed to be is very low. But when the signal molecule binds, it causes a shape change. The shape change uses some energy to activate some other proteins, right? Step one, step two, step three, step four. It's still the same pattern. Reception causes shape change, causes the next protein to be activated. The ion channel doesn't follow that pattern exactly because this is a very simple idea. This is just a doorway, right? When there's no signal molecule, the gate is closed. The molecule binds, which makes the gate open, and things can go through the tunnel. When you want the gate closed, the signal molecule falls off and the gate's closed, right? So this is still a conformational change, but this one isn't as complicated as the others, and because it's just opening a door. So we've talked about signal molecules. We talked about protein receptors. Here are two questions for you to think about. If you have a water-soluble signal molecule, where would you find the receptor? And then what is the common theme for all protein receptors? If you have a water-soluble signal molecule, you would expect the receptor to be membrane-bound because water-soluble means polar. I wanted to make sure you remember that, right? And polar molecules cannot go across the cell membrane, and therefore the receptor has to be on the outside of the cell membrane. What is the common theme for all protein receptors? It is shape change. Conformational change is the scientific way of saying that. But again, this is a pattern that we've seen a lot of times already. Now, to transduction. Transduction is the most complicated part, but again, you won't have to remember too many specifics, mostly looking for patterns in the way that the relay molecules are triggered. So, starting with the basics again, the basics means that we are going to have a cascade of relay molecules. Usually this is called the phosphorylation cascade. It's not always that. We'll see that in a second. But the idea would be that the first molecule triggers the second molecule, triggers the third molecule, and so forth, right? This is going to be the pattern that we're going to see the whole time. Molecule 1 makes molecule 2, makes molecule 3, and just keeps going and going down the line, like dominoes. Now, there are these things called second messengers, which are not phosphorylating things, but we'll get to those in just a moment. Let's start with the phosphorylation cascade. First, phosphorylation means adding a phosphate group. We're going to put a P onto some sort of molecule, and that molecule is going to be activated. The name for the proteins that do that are called kinases. This is a word that you end up seeing a lot. In my experience, this word kinase gets used most commonly where they don't tell you what it means, right? A lot of times they'll give you a vocabulary word, but they'll kind of explain it at the same time. But for whatever reason, kinases are one that you definitely need to remember. Now, kinases add phosphate groups. To turn off the protein, we have to dephosphorylate it. We have to remove the phosphate group, right? This is done by something called a phosphatase. Again, I don't know exactly why, but this word usually I don't see on the AP exam, and when I do, it is being told to you what it does. So remember the kinase for sure. It would be nice to remember the phosphatase part, but phosphatase is for turning off the signal cascade. So usually that's not the like subject of a question. Usually we want to know about what happens when you turn something on, right? So that's just a real life practical implication of all this kind of thing. So here is a picture of a phosphorylation cascade, and it is rather complicated, as you can see. This is definitely one of those times where you need to remember that you should not be looking at this as if it is one picture. This is 9 pictures combined into one picture, and you should be looking at it that way, and that's the way that I'm going to look at it right now, right? And so I want to start at the beginning, and I'm only going to focus right there, right? So, of course, on my screen, I can make the other parts go blank, but when I'm looking at this, my eyes are only focused here, and I'm not paying attention to any of this other nonsense down there, right? Because it's only going to make things complicated and confuse me. So step 1 is reception. We know that. Reception usually has some sort of activated relay molecule. Maybe that's the G protein, right? Now notice that activated relay molecule is not a kinase. It's not adding a phosphate to anything. It's just turning on the next protein. How does it do that? I don't know. There's probably I know that there are a variety of mechanisms that you don't need to know about. It just happens in a certain way. Now, once you have the activated relay molecule, its job is to activate something, right? Just like all of these, the pattern is molecule 1 activates molecule 2. So this time we are going to activate a kinase, right? So notice active protein kinase. Its job is to turn on the next protein in the chain. It's going to do that by this time for sure, adding a phosphate group. Where did that phosphate group come from? From ATP, right? ATP is really our source of phosphate. It makes everything happen, right? And on that note, it's important to notice that this does require energy, right? If you want to carry a signal throughout your cell, it requires energy. So just sitting there, not doing anything, the cell is using ATP. Now, active protein kinase 1, it has successfully activated kinase 2. What is kinase 2 going to do? Same thing kinase 1 did. Add a phosphate to another protein in the chain. Where's that phosphate come from? From the ATP, right? So that's just going to do exactly what it did before. Notice these 2 pictures are basically the same, right? It's a repeat of the same step. That step could be repeated numerous times. Depends on what the cell needs. Now, active protein kinase 3, what is it going to do? It's going to do the same thing kinases always do. It's going to add a phosphate to something. This time, it's going to add it to this pink protein, which isn't pink in real life, of course. Now, this pink protein in this diagram is what's going to go off to do the response. This is the thing that's going to trigger whatever we want to happen, right? Now, it made it again. It's at the end of the third kinase. It doesn't have to be 3. You could have 7 in a row. You could have only 2. It doesn't matter how many you have. It's just this domino effect. So going back to look now, so this is the way that my brain would work. I would look at each part specifically, and then I would start to look at it a little bit more, right? So I'm not going to try to look at the whole thing. Now I'm just going to try to put all this together in my mind, right? You can clearly see the domino effect. Trigger 1, triggers number 2, 3, 4, and just keeps going, right? The domino effect is happening. Now, at some point, this active protein will have done its job, and the cell doesn't want it to keep doing its job, right? Like we have successfully done the response. We have to turn it off. This means we have to take away the phosphate group, and there's our phosphatase, right? Notice that each of these steps is showing that you can also inactivate it by removing the phosphate. The phosphatase is taking away the P, right? Now, of course, these little phosphates, inorganic phosphates, those would fly off to go find an ATP synthase so that they can get put back onto the ADP to make ATP. So again, now that we've broken down the picture, we can kind of look at it in one big shot. But again, I want to point out some things, especially about this diagram, and that in this diagram, the arrows are not exactly showing movement. They kind of are showing movement, but really the arrows are showing time. The arrows are showing this is how it started and this is how it ends up. It didn't move from one place to the next. It just changed its shape, which is why these diagrams aren't super helpful in this case, because these are static diagrams, right? They can't show the changing of shapes. They have to show the two different shapes. So one other thing to notice is that while this looks like it has 8 proteins in it, there's only 4 proteins that are actually involved in this whole process. But it's being shown twice because they're trying to show time. So again, this is where static diagrams kind of break down, and not to spoil too many things, but you're going to make a dynamic model of this very soon. So the last thing I wanted to point out again is that this is a dynamic process, that when you go back and look at this, maybe by the time this protein has been activated, the first kinase maybe is already deactivated. And maybe even the second one is being deactivated. Maybe this protein phosphatase step has already happened while this part is still happening, right? This is not this like in real life like dominoes where you have to knock them all down and then, all right, they're all knocked down. Let's set them all back up again slowly, right? You would imagine as the dominoes are being knocked down, you are behind it, setting them back up again so they can be knocked down again immediately. Right? And so it's really about equilibrium. It's about the balance of how many active versus inactive proteins you have going on. It's very complicated, very much the mosh pit that I like to think of it as. Of course, I'm not going to give you any, in the AP, wouldn't give you any questions that requires you to really think of it in the dynamic way, but just so that you realize the activity that's happening in a cell, that these pictures make it seem very simple and straightforward, where it is even more complicated than it actually is. Now, that's the phosphorylation cascade. We don't always have to use proteins in the cascade. In the relay race that we are running, most of the time they are proteins, but sometimes we can use small polar molecules or ions. For example, something called cyclic AMP is one that we will use, or calcium ions, right? Cyclic AMP is a molecule, calcium is an ion. Why would we do this? We do this because we don't have to turn on the second messengers. They are just little tiny ions in some cases, right? Like you can't activate calcium. Like it's just a little, a little atom that's missing a couple of electrons. So they don't need to be turned on. They can just do their job immediately. And you can make a lot of them. This is going to be the big thing. And we'll talk about this actually a little bit more later on. But instead of one protein activating one other protein, maybe one protein is going to cause 100 second messengers to come out. And now you have spread your signal 100 times instead of only once, right? And so this, you could see maybe that would have a benefit. And we'll see some examples in a minute. Now, cyclic AMP, what is cyclic AMP? It's this one right here, and it's called it because you might notice that this carbon is connected to the oxygen, and that oxygen is connected to that carbon, right? So it makes a loop right in here. Now, that has to be made from ATP. right? We are actually, in fact, going to use energy to make these molecules, and that requires an enzyme called adenylyl cyclase. Adenylyl cyclase happens to be right there in this example, right? It doesn't have to be there, but it is found there very often. This is a common cascade. So the first messenger, the signal molecule, triggers the receptor, which triggers the G protein, which triggers the adenylyl cyclase, which is then going to make a whole bunch of cyclic AMP, lots of cyclic AMP. And then those are going to go off to activate things. In this case, here's our first kinase, right? So even thinking back to the picture that you just saw before about the phosphorylation cascade, it went from the receptor to the G protein directly to the kinase. This time, there's a step in between G protein, AMP, then protein kinase, right? So again, don't get locked into like this is how it's supposed to be. All that matters is that step 1 triggers step 2, triggers step 3, triggers step 4, triggers step 5, and so forth. These are all very mix and match, right? Sometimes the cyclic AMP will happen in the beginning. Sometimes it'll be in the middle. It could be the last thing that gets triggered before you then get the response, right? Like it could be anywhere. This is all available for the cell to mix and match depending on what the cell needs to do. And again, that's all based on DNA, and that's all based on evolution. Calcium is another second messenger. And again, you don't have to memorize what calcium does or any of the things on this picture, but I do like this picture because it illustrates what I was just saying, that there are a lot of different variations on the same pattern. Here we have our first messenger hitting our receptor, hitting our G protein, hitting another protein called phospholipase C. I don't know what it does. It apparently triggers this molecule to turn into this molecule called IP3, which, as you can see, is another second messenger. So we are now seeing a cascade that kind of involved the protein that involves then a second messenger. This is going to hit another protein receptor, a gated ion channel, to release another second messenger called calcium. Calcium's going to go off to do lots of different things, right? And this is the benefit of second messengers. A kinase can only go and phosphorylate its one target. But calcium can go off and come into contact with all kinds of different molecules. And those molecules would be needed for whatever response is going to happen. So again, you'll never have to memorize this picture that's on the screen. If you were going to have to talk about it, they would give you this example. And your job is just to recognize the pattern and to recognize that sometimes they're proteins and sometimes they're second messengers. Yeah? So again, the last one we didn't really talk about is the gated ion channel. So just real quickly, the cell spends a lot of energy to keep the calcium inside the endoplasmic reticulum or inside of lysosomes or other vesicles. But you don't really want calcium just floating around because calcium is a second messenger, right? So most of the time in the cytoplasm, you would expect there to not be a bunch of calcium floating around. But then when you need the message, you have to open the gate, right? And so first, when the cell puts all the calcium into the endoplasmic reticulum, would this be an example of active or passive transport? Going back to some old things. Of course, it's active because it spends energy, right? Then those calciums are now in the area of high concentration, and when you open the gate, they're going to diffuse outwards, right? And so again, I just want to show a close-up of this example of how this is a little bit different than all the other receptors that when you see this happening, that's really this part being shown, right? And so again, if you were going to memorize one of them, it would be this one, the gated ion channel, because it is so straightforward. But again, you shouldn't have to. It would always be explained. Right? So this is just another way to have the second messengers. So here, what are the two ways? There's two different strategies that can be used to transduce the signal. And then what types of proteins are part of that transduction cascade? Phosphorylation cascade is one way that we can transduce a signal. Second messengers are the other way that we could do that. Remember, second messengers, there's various ones, but cyclic AMP and calcium are the ones that we talked about just now. What types of proteins are part of the transduction cascade? If they are a protein, it would be a kinase, and its job is to add a phosphate group to another molecule, right? And so in the end, you have your relay race of molecules connecting to the next molecule, which triggers them to connect to the next molecule. And again, I'm saying this all very vague because you can take all of these different things and mix and match them into whichever way that the cell needs them to be mixed and matched. This is then going to make the response, but I don't want to keep adding more things on, so there'll be a separate video for the response.